The nucleus of each cell contains a complex of macromolecules called chromatin. Chromatin is a densely compacted form of DNA double helices, wrapped around histones. All of the genetic information defining the proteins of an organism is encoded in the DNA structure of chromatin. The helical backbones of DNA are a phosphate deoxyribose. These backbones are connected by a base pair of nucleic acids. DNA is composed of an ordered string of 4 different bases: adenine, thymine, guanine and cytosine, referred to as A, T, G and C. Two, and only two, combinations of nucleic bases exist in DNA: AT, or TA, and GC, or CG. These two combinations of 4 bases are the sole alphabet of gene expression. To enable gene expression, a fragment of the DNA double helix must be temporarily unwound and separated into 2 individual strands. The nucleic bases contained in this single chain of DNA determine the protein to be synthesized. During transcription, a segment of DNA is copied into mRNA by the enzyme RNA polymerase. Transcription produces a complementary and anti-parallel mRNA strand called a primary transcript. mRNA consists of the nucleic bases adenine, uracil, guanine and cytosine, A, U, G and C. Translation. During translation, ribosomes in the cytoplasm organize chains of amino acids by decoding the mRNA chain. A triplet of 3 consecutive bases, called a codon, is read by the ribosome. A tRNA molecule, charged with the fitting amino acid, docks onto the ribosome and the amino acid is added to the polypeptide chain. The ribosome then progresses to read the subsequent codon. This process is repeated until a stop codon is encountered. Translation is then terminated. Polymerization. During translation, the individual amino acids of the polypeptide chain are polymerized to form a simple protein. A single protein consists of 20 to many thousands of amino acids. Summary. DNA in the nucleus contains all genetic data. An mRNA chain is the complementary to the DNA. Triplet codons of mRNA bases determine amino acids. Amino acids are polymerized to form proteins.